Welcome everybody once again to the second wave of Coffee with EMBL. It's the second wave in every sense. I think this is the second session this year when we've restarted Coffee with EMBL sessions, and it's the second set of Coffee with EMBL after the summer break. Sadly, there may be other types of second wave going on, which I'm sure everyone who joined in is concerned about. That's one of the topics we're going to get on to discuss. I think it's at the front of all of our minds. It was absolutely fantastic when Ush said that so many people had registered for the meeting. Probably some people will be joining at different times as we go along, but I think we've had 180 or so people register for the meeting. Fantastic, again, with representation from countries all around the world, from most continents. Really fantastic opportunity for people to connect and for us to exchange information about how the situation is developing in their own countries. It's also, I think, really great that as the Coffee with EMBL evolves, that we see clearly that it's an event both for EMBL staff, or as I like to think of them, pre-alumni, and also for alumni who were formerly EMBL staff, and not only scientists, professional scientists, but people with a whole range of interests. Some people who are scientists and have moved into education, broadcasting, the media, people who are lawyers, administrators, interested in other ways. This is a celebration of a diverse family of EMBL staff and alumni, and that's just fantastic. Something else that I think most of us will have seen in the last week or so was the most recent awards of Nobel Prizes, and we'd like to congratulate everyone who won a Nobel Prize this year. And I have a shout out to Emmanuel Charpentier, who is an alum from one of the Nordic partner institutes of EMBL. So fantastic. I think we were all expecting to see CRISPR rewarded with a Nobel Prize. Great to see that happening. And also, it's very much in line with the theme of today's meeting that there was also a Nobel Prize given and shared between three prize winners for their pioneering work on detecting and characterizing hepatitis C virus. And that's going to be a theme we'll come back to in just a second with one of our guest speakers today, Raffaele de Francesco. Raffaele is the 2018 Lennart Philipson Award winner, as many of you will know. I know him also as someone who worked at EMBL a long time ago, the same time as I was there in the late 1980s, 1990. And he is famous for his work on hepatitis C virus and the development of medicines to treat that. Interestingly, I'm sure Raphael will tell us more about this in a few moments, but as we're all promised that there'll be vaccines to save us from coronavirus, it's worth bearing in mind, I think, that after more than 30 years, we're still waiting for an effective vaccine for hepatitis C virus, along with many other pathogenic viruses that affect us. But it has been possible to develop very effective antivirals, and that may be an alternative route to getting us out of the current problems we're having. So we very intentionally took the opportunity to call this second wave. It's something, it's a bit scary. Mind you, if you think this is scary, the next event we're going to have will be on Friday, the 13th of November. So that's also a scary date, and we'll see whether things have developed positively or not with respect to treatment of the coronavirus pandemic. We want this to be a highly interactive event. We really encourage people to put comments in the chat, and we'll try to bring in as many people as possible. As in previous events, in addition to the main event, during the first hour, we'll extend for a second cup. And we hope during that extended period to bring in as many people who'd like to make a contribution to any of the themes or comments that we've discussed. The very first of these Coffees with EMBL really started off when we went into lockdown together in Europe back in April. And we wanted to bring the community together, learn more about what was happening to us, what the science was behind coronavirus and the pandemic, what the health implications were, how we were all coping with this, and how our different countries were responding. We had a series of events back in the springtime and early summer focused on this. I have to say, it had been our sincere hope that without ignoring that by now, that would no longer be at the forefront of our thoughts, and it would have been something that we had learned from and moved on. That doesn't seem to be the case. And after the numbers of infections, certainly across Europe, falling down, in many countries, the numbers are rising again steeply. And different countries are seeing different trajectories of the infection. So we wanted to just ask people, since we have people online from so many different countries, if you could let us know, are you seeing a second wave in your own location? And if you just like to open up the participant group, and at the bottom, there's a yes and no buttons. I'm going to click yes, because certainly in the UK, that's certainly happening. So it'll be interesting to see from the locations people are in, if they feel that a second wave is already taking place. So one person is not seeing a second wave. That may be Toby, for all I know, but or it may genuinely two people. So that must be Graham Tebb as well. But there may well be locations, since we have people from so many different places. But overwhelmingly, people. I mean, so 34 people who have voted have said yes, they are, and I think that's just the reality of the situation, and so it does make us wonder. We've already been through incredibly severe lockdown restrictions in many countries throughout the world. Most of us have experienced a profound change in our way of working, our way of life. Our freedom to meet with family and friends has been curtailed as we attempt to respond to this. I think we need to look for some way out of it. And so we need to turn to people who have been studying pathogenic viruses. And so I'd like to bring in Raffaele.
Hi there. So this is, my name is Raffaele de Francesco. I was at the EMBL between 88 and 1991, actually. And after that, I moved back to Italy, where I started to work on hepatitis C. And I worked on that virus for around 25 years, actually. So I'm very glad that the hepatitis C community was recognized by, as a whole, I think, by this Nobel Prize award that was given to Orville Alter, Michael Houghton, and Charles Rice for the discovery of a virus, which I can, briefly summarize. So basically, Orville Alter was at NIH, and he was actually the person who recognized that there was a third agent that would cause infectious hepatitis from infected blood. So he coined the term non-A, non-B hepatitis, and later Michael Houghton, he was at Chiron Corporation in California at the time, he's British, discovered a clone, which is the sequence of a virus. It turned out to be a positive-stranded RNA virus, just like coronaviruses, if you like, that is more a relative of flaviviruses like Zika, for example, dengue, yellow fever. So for many years, HCV remained just a sequence, a clone, until Charlie Rice was actually able to construct an infectious molecular clone, injected an RNA derived from that cDNA clone into the liver of a chimp, and demonstrated that single agent was enough to give hepatitis C. So basically, it closed on the Koch postulates, if you like, showing that, not only there was an association, but it was actually a causative agent of hepatitis C. So very good for that. And then after the discovery, however, it took, as I said, 25 years to actually develop effective antivirals. Now there are very effective antivirals on the market, and within a few weeks, taking one pill a day, people who are chronically infected with the virus can actually get rid of the infection and clear the virus from their body in a permanent way, which is very good news. So actually, what Angus mentioned already, I think, is there is no vaccine for hepatitis C yet. Certainly, research in hepatitis C did not progress as fast as we are witnessing for SARS-CoV-2. But still, there were several candidates, several clinical trials, and nothing that worked was really coming up. And so we still have no vaccine. So this actually, it's very relevant to a major question, and that is we only have eradicated one virus now from the planet, and that is smallpox, and it was eradicated because of vaccine. We are eradicating. We are on the way to eradicating poliovirus, and that is because of the vaccine. And now for hepatitis C, there is the opportunity to actually eradicate a virus with antivirals. So that is a realistic possibility, but it's still a pretty winding and long road to go. So, but the question is, why don't we have a vaccine? We don't have a vaccine because the main reason I was told by immunologists is that, basically you can get infected with hepatitis C and recovery from the infection spontaneously, and then you can get reinfected again. And actually drug addicts get infected multiple times. So there is no natural sterilizing immunity against hepatitis C. And this is one of the reasons why it is so difficult to get a vaccine, combined with the fact that there is a lot of variability. The virus replicates owing to the action of an RNA-dependent RNA polymerase, which does not have any prophylactic mechanism. So there is a lot of genetic variability, which makes it difficult for a vaccine to be developed. So in that respect, there are several considerations that one could make when thinking of this pandemic that is going on right now. And one is, will we have a vaccine? Well, I hope so. Certainly, one thing is in our favor if you compare to hepatitis C, and that is the variability, the intrinsic variability of the genome of coronaviruses is not as extensive as for hepatitis C. And that is, the reason for that is actually the replicase, the polymerase of coronaviruses as a proofreading mechanism. The genome is very large. It's actually the largest RNA genome on the planet, a known RNA genome. And to actually maintain such a large RNA genome, a proofreading activity is required. So coronaviruses are much more stable, and there is much less of variation at the genetic level compared, for example, to HIV or hepatitis C. And that is good news. More sobering is the fact that also for coronavirus, like it is the case for hepatitis C, you can get infected more than once in your lifetime, and even within probably a short period of time. So how protective a vaccine will be will remain to be seen. Now, the another consideration that I want to make is that with respect to time, so it took 25 years from virus discovery to effective drugs for hepatitis C. Obviously, we don't have that type of time. I mean, hepatitis C is a chronic infection. So it's there. It's going to, without human intervention, would stay there forever. SARS-CoV-19 is an acute disease. So, either you recover or you die. And we don't expect the virus to be around for more than 1.5 more years, I believe. So we have to hurry. I mean, so will we able to develop an antiviral, and is an antiviral needed? I think an antiviral would be great, and would be great to have a broad spectrum coronavirus antiviral. And I say this because I expect that more coronavirus pandemic could happen in the future. So essentially after SARS-1, everybody stopped working on coronaviruses because it was over in a very short time. Now we're going back to antiviral candidates that were developed for SARS-1, hoping they work on SARS-2. But I think now we don't have to stop. We have to keep working on this family of coronaviruses, possibly other families as well, regardless of the pandemic being active or not. Essentially, we need to stockpile antivirals for future emergency.
Raffaele, there's a question that's come in from the audience, and we encourage people to put questions in the chat. So from Margaret Burmeister and Ann Arbour, I was just asking if you could comment on the reports in the literature or the media, in fact, that there are some, admittedly rare, but some apparently validated cases where people have been reinfected with coronavirus, where the second infection, the phenotype, the severity was worse. Could you comment on what mechanism might be involved, or why is that so rare, or is it something seen with other viruses?
Well, with common cold coronaviruses, actually the immunity can last anywhere from weeks to months, and usually wanes in about a year. So, but there is a lot of variability. So, immunity to coronavirus, natural immunity to coronavirus is not, it doesn't last very long. Like it is the case for flu as well. I mean, for flu, immunity does not last very long, also because the virus changes. In this case, the virus is more stable, so it's an intrinsic property of our immune system, probably not to have a very long-term memory, at least in terms of antibodies. But there could be cellular immune response, which may last longer and protect, if not giving sterilizing immunity, could give some level of protection. That is, I mean, it's just something that remains to be seen in larger numbers.
So what could you share with the audience about the confidence in the delivery of either effective vaccines or antivirals in a time frame that's sufficient to address the current pandemic? As you said, it may be based on historical pandemics that within 6 months, 1 year, 2 years, the virus will no longer be as serious a problem. Although it would be interesting to know if you feel that's true. But what's the chance of medical intervention getting us out of this current situation?
Right. So I think, developing an antiviral, unfortunately, I think will take longer than 1 year from now. So there are basically now 2 major classes of antivirals that are in development. These are polymerase inhibitors, or nucleoside analogs, as well as protease inhibitors. It will take there are a few compounds which have been in clinical trial already, but gave disappointing results. So I think it's going to take some time, and I think those compounds are going to be, those drugs eventually are going to be useful to have also in view of possible future pandemics. But I honestly doubt they're going to be very useful this time around. There is also to say that actually to use an antiviral, you actually, or even a monoclonal antibody, which is a type of antiviral, if you like, you have to really be able to dose it in the viral phase of a disease, which lasts only about a week. So an antiviral will not work after a week after you get infected, when actually the immune system response kicks in and when the cytokine storm begins. So that leads me to think that what we really need is faster tests for diagnostic tests. Now it takes too long, and we need more frequent tests. I think that's something we need very, very badly to contain these epidemics. Tests, antigenic tests, possibly that you can run very quickly and very frequently to track the disease, together with distancing and mask and everything else. A vaccine, I think, may be developed in right time. Let's put it this way. I think a vaccine could be approved in a relatively short time. Now, manufacturing it and vaccinating everybody will require a long time.
So I don't want to put you on the spot. Actually, yes, I do. I want to put you on the spot, Raffaele. But then also open it up for everyone else. Coronavirus isn't the first either pandemic in our lifetimes. It probably is for everyone here. But certainly in all of our lifetimes, there are other epidemics that have happened in many countries that have had high mortality rates. And as we will come to discuss a little bit later, there are many other types of infections and morbidities that also have very debilitating effects on populations. Do you feel that we are overreacting as a society, that we were just so unprepared for this, that we are overreacting to the reality of the situation?
I don't think we are overreacting, and this feeling. I say this because I witnessed what happened in Italy, and what was really scary was the intensity. So basically, Italy had relatively few infection foci, if you like, outbreaks, and a lot of people died, but despite that, there were local outbreaks. So if you think of something like that happening in a nationwide or global scale, it'd be very, very scary. So I don't think we overreacted. I think the lockdowns were the right thing to do. Right now, I'm not sure any longer that we have to that lockdowns are a solution anymore. Because actually, as you said, now mortality, real life mortality has decreased a lot. In Italy, we're beyond 10%, but now we know that fatality rates, infection rates, is more in the order of 1% or less, which is much worse than flu, but not as devastating as it appeared initially. So I think really we have to learn how to live with this virus. So be very cautious, but I'm not in favor of additional lockdowns personally. I think we have to test more than anything else. And I don't think we can lock down again, thinking that we will have a vaccine in 2 months from now. That's not going to happen. I don't think so. I think maybe vaccine will get approved and some people will get vaccinated, but it's not realistic to think that everybody will get vaccinated by the end of this winter. I don't think that's realistic.
I tend to agree with you from my understanding and interaction with colleagues that I think it's unlikely that science alone in terms of therapeutics or vaccines will save us from the societal impacts in the short term. But I'm sure there's a lot of opinions out there, and please contribute in the chat. And I'm sure that's a theme we can pick up and talk about as the discussion goes on. Again, coronavirus is not only not the sole pathogenic virus, as we all know. It wasn't even the virus that was expected to be the cause of a pandemic. I think most people who talked about this, and I had certainly attended myself, virology and microbiology conferences where people speculated where the next pandemic would come from. And I think most people were looking at influenza as the most likely cause. I'd like to bring in Jan Kosinski from EMBL in Hamburg. As a young group leader working there, Jan started his EMBL career in Heidelberg, but is now a group leader in Hamburg, where he's studying nuclear pore complexes, and has also moved his research interests into infection biology. And when we had originally invited Jan, it was very much with the thinking that we moved on from coronavirus and we're going to think more broadly about infection virology. And I can't help reflecting the field of molecular biology, really so much of it was learned from studies on model systems that used viruses of different sorts, all the way from bacterial viruses like Lambda up through adenovirus. I was a postdoc many years ago at MIT with Phil Sharp, who won the Nobel Prize for discovering splicing by studying adenovirus. And so there's a rich history, even for people who are not virologists or don't see themselves as virologists or studying medical implications of infection directly, to have learned basic features of cell and molecular biology by studying viral systems. But Jan, you have made the decision to move into studying infection biology. And do you think you're going to help us meet the next pandemic?
Yeah, hopefully. Actually, yes, the story goes like, definitely I don't have as much experience as Raffaele has with viruses. It's just 2 or 3 years we are working on the influenza virus. And definitely before we haven't worked so much, or I haven't worked so much on this subject. So we're focusing on protein structures of different protein complexes, and the key motivation was that we are able. So I work mostly computationally, and we were able to predict based on our models, computational models, predict how the complex could work based on very fuzzy data. And the key motivation for this is, can we do it for processes? And we actually took influenza virus as a model system, not actually with an intention to study infection biology, but to basically study dynamic processes. And I thought at the time that infection cycles actually is a simple case of a process to study, because it has like at least cellular infection cycle has a definite starting point, and the entry to the cell and the exit. That time I thought it's that simple. And I guess Raffaele can confirm it's not that simple in the end. So it's a very complex processes, and especially for influenza, you have many different strains. So the data that you obtain when you study these viruses is very heterogeneous. Data obtained for different strains in different labs can be conflicting. So we are focusing on basic research actually to understand how this virus replicates within the cell. Actually focusing what it does in the nucleus, how it exits the nucleus. But in the same time, we want to use it as a model to develop methods that can hopefully, in addition to help us to prioritize what pathways that are involved in infection, or which proteins are the most crucial for the virus and are most interesting to study experimentally, but maybe also as targets for drug development and development for antivirals that target host proteins. So who knows? Yeah, maybe if coming back to your question, maybe in the end we can somehow provide models or testable models of hypotheses that can help in preventing the next influenza pandemic. And maybe also we can develop methods that can be used for other viruses. So as well.
So one question that comes from that, and we'll come back in just a second, but actually we want to have a little poll with everyone. I think Peter's arranged that you can respond to this poll and hopefully see it on the screen, is as we try to think about how science can contribute to better understanding the trajectory of the infection, how we should respond to it, perhaps creating therapeutics to treat the virus. Should we all, who are still working as practicing scientists, should we all switch our research focus and, for the time being, all of us just start studying coronavirus, or not?
Can you see the results, Angus?
I can see it, yes.
Okay.
I can see it. I've voted, so people are voting. That's great. Hopefully you can see it on the screen. So, yeah, and again, to put you on the spot, do you feel under any pressure to say, okay, I should really now start to work on coronavirus because I could make the difference?
Yeah, definitely. So actually, we feel that maybe that's the wise thing to do, because influenza is also a very dangerous virus. But if you look at the statistics at the moment, even if you take the most pessimistic assessments of mortality or deaths per year from influenza, I think the highest estimates are like in the numbers of like few hundreds of thousands. It's still less severe than COVID. So maybe we should actually switch. On the other hand, we are many of the research groups are small with limited resources, with PhD students and postdocs with fixed term contracts. Can we now change the projects for a year? Then it may be difficult for the careers of the people. So we haven't actually switched yet or started a collaborative project. So we kind of resisting that. But it's mostly for resources, and maybe thinking also, actually, if we just start jumping now from virus to virus, what happens, I think the second wave of the coronavirus is the last, and then the influenza comes next year.
And I think,
I think that's correct. Raffaele, please come in.
Yeah, we should. You were addressing this point earlier, but let's remember that still about 1 million people every year die for hepatitis of viral origin. Eventually they die because of liver failure or liver cancer, but hepatitis is the underlying cause of viral hepatitis, and then another million of HIV. So there are. We have to work more on coronavirus, but we don't have to really stop working on other infectious diseases. That's my point.
Yeah, no, I think the majority of people who responded to the poll agree with that, in that 77% of people said no. And so infectious disease, again, it isn't the only cause of morbidity. There are other illnesses, and I was having a discussion with some people before this started. And in a conversation last week with a collaborator from the medical school, who is the chief cardiologist in the local area, he said locally in Scotland, he's never seen in 30 years so many people dying from heart attacks, not in hospital. And that's an indirect effect of the fear of infection and the coronavirus, discouraging people who think they maybe only have mild symptoms, not responding to, not reporting, or not being taken into hospital and treated because it's seen as not threatening, and they go into cardiac arrest. And so there's almost an epidemic of secondary indirect effects of the fear of the virus spreading. But then, as well as pandemics, there are also what you correctly reminded me, Raphael, are endemics, which are the terrible effect of many types of disease, including parasitic diseases, which affect many parts of the world, perhaps less so in the Western world or in Europe, but which still affect the lives and cause large numbers of deaths. And so I'd like to bring in, at this point, Maria Bernabeu from Barcelona, who is a new group leader there. Also, congratulations to Maria on receiving a prestigious ERC grant recently. And Maria works on cerebral malaria. And I think I'm going to hand over to Maria because it's important for us all to remember that coronavirus absolutely is not the only disease. Maria.
Thank you, Angus. Hello, everyone. So I started my own group in malaria about a year ago at EMBL Barcelona. And I've been working with malaria through all my scientific career. I did my PhD in Plasmodium vivax malaria, and then I moved to Seattle after that to study Plasmodium falciparum malaria, which is one of the deadliest, is the deadliest malaria species. So I'm going to share my screen now. And since I'm an EMBL, so I've always worked in infectious diseases centers. And since I'm an EMBL, I always receive this question, why are you studying malaria? Why are you doing that? I never had this question before because I used to network with parasitologists or infectious disease biologists. So I usually always start my presentation showing the map where malaria is endemic, where Plasmodium falciparum is endemic, and showing the numbers. So there's every year about 200 million clinical cases per year, and there's about 500,000 deaths per year. And I have the impression that every time I say these numbers, people receive it cold. So they are big numbers, but you don't really think how severe this is until you compare it with COVID. We've been living with COVID for over a year now, 9 months. Maybe we don't know when it was originated, but there's been 39 million cases reported. Probably this number is much higher because we did not have good diagnostics on the first wave. And there's 1 million of deaths that have been reported, and this number is pretty accurate. And if you compare it to 1 million deaths with 500,000 deaths from malaria, then you realize how big is the malaria problem. And this has been happening forever. We have lived with malaria since there are humans, before we separated from primates to humans. So there's always been this super big burden on malaria. And we don't hear back in the news because it's affecting people in Africa and it's affecting people in South America, South Asia. And it's affecting every year. It's not an outbreak, a pandemic like now. So that's one of the reasons that attracted me to malaria. Second reason, of course, is that it's a fascinating. Parasites are fascinating, and they are super complex and unique. So that's what really kept me wanted to work in malaria. And then, so this 500,000 people dying. So why are they dying? So there's many complications that malaria patients can happen. But in kids, mostly the most fatal and pathogenic complication is cerebral malaria. So you can see this super striking image of one kid with uncomplicated malaria on the left and one kid with cerebral malaria on the right. And without being a radiologist and neuroradiologist, you can really see there's something wrong with this brain. There's this massive brain swelling that these kids have. And this massive brain swelling, because you have the school, what happens is that the brain cannot expand more, and then it starts compressing the brain stem. So these kids end up dying because they stop breathing. So we have the respiratory center here. And these kids are in rural areas and stop breathing because of disease. And this happens because in malaria-infected red blood cells usually try to escape spleen clearance because they are very rigid. So Plasmodium falciparum developed this strategy of sequestering in the microvasculature. So it expresses an antigen that binds to endothelial cells. And if these kids have huge parasite biomass, this sequestration will happen everywhere. But when it happens in the brain, it's especially pathogenic. And you can see in this image where we have each dot is a malaria parasite. It's not circulating, it's stuck here, and it's causing microvascular obstruction. And this leads to this blood-brain barrier breakdown and massive brain swelling. And this is what we're studying in the lab. We are very interested to understand how we go from microvascular obstruction to this massive brain swelling. And by looking at patients and humans, we don't have a lot of clues of what's going on. So we can see on MRI, there's this massive brain swelling. We don't have resolution to know what happens at the molecular or cellular level. We also can look at autopsy studies like this image that I'm showing you here, but this not explains what the progression of the disease, what was the molecular pathways that led to that. There are no good rodent malaria models. Rodent malaria models do not sequester and brain microvasculature at all. So it's not a good model to study. We can use it to study cytokine storm and how pro-inflammatory pathways cause disease, but not how parasites cause disease. This is what we are doing at EMBL. So our lab, we are building 3D engineered microvessels. So in this video, you can see one of our capillaries that we built in Seattle, and you can see our beautiful system that can be perfused with blood at regular hematocrit. And I love this video because you can see this fascinating blood microfluidics here on the top. I'm going to stop this one of the blood, red blood cells accumulating in this pre-arterial region, and then how they go through this capillary one at a time, and then how they leave the capillary into post-capillary venule. And we can use this system and perfuse it with malaria parasites. And you can see here one of our strains that cause cerebral malaria, how infected red blood cells sequester in this capillary region and create this microvascular obstruction. So this model that I'm showing here is in 3D, 3 dimensions. We build it with only endothelial cells. So the goal in the lab will be to build this model with a blood-brain barrier-like So we will add other cell types. We will start with pericytes and astrocytes, which have been shown to increase endothelial barrier properties. And in the future, hopefully, we can also introduce neurons into the model and see how Plasmodium falciparum affects endothelial cells, but also other brain cells. We can also build organ-specific vasculature, so you can model other parts of the malaria cycle. You can model skin infection, liver infection before parasites reach the blood. We can also use this system to study other diseases, but the main focus of the lab will continue being cerebral malaria.
Thanks a lot, Maria. So, I mean, that was a remarkably clear demonstration of just how many people can die from diseases that are not coronavirus, and also a reminder that even after hundreds of years of humans living with this, and while I understand you from the map you showed that the vast majority of people affected no longer are in Europe, but it wasn't so long ago, and this was brought up also in the chat that many European countries were also affected by malaria not so long ago, and yet we have no vaccine, we have no really good anti-malarial compounds. Is that just a consequence of the amount of resource being devoted to study this disease? And could it be that actually the current pandemic will even suck more resource away from malaria, from the countries? Is that an even bigger risk of another indirect effect of the pandemic, of making the study of diseases such as malaria even harder?
Yeah, so you touch many points here. So going back to your first point, yes, there was malaria in Europe. In the 20s, 30s, I remember my grandparents mentioning friends having malaria. And then we got rid of mostly by using insecticides. Then the species that was affecting Europe, it was mostly vivax, which is less severe, but that's the reason, for example, why the CDC was funded. So the CDC, Center of Disease Control, it's based in Georgia, which was one of the major endemic sites for malaria in the US. So that's one of the main reasons the CDC exists right now, because it was funded to study malaria. And then we don't have malaria anymore in Europe or the US, and so now it's mostly in tropical areas. And we have very good antimalarials. So you can use combination of artemisinin-based compounds, and they are very good. So they clear parasites from blood very, very quickly. So in 24 hours, patients rarely have parasites in blood. And what we've seen with these cerebral malaria patients is that sometimes if you just let the antimalarials work, patients will recover. So these patients recover in 24, 48 hours, very, very quickly. But the problem is that you have these patients that have this very acute brain swelling, and you need to treat them right now. So antimalarials are very good, and there are campaigns of intermittent treatment in kids that have been working very well. About 10 years ago, 15 years, malaria mortality was twice as much because there were not as many resources being used to treat this disease. So with this increase in funding of malaria prevention, we've reduced the number of deaths to half the number we used to have. Yes, there's a worry that with COVID, we don't funds to malaria dedicated to COVID. What I've been seeing from my colleagues working in Africa is that it seems that African countries still want to keep this funding dedicated to malaria. So hopefully this won't happen this year or next year. So yes, I think COVID might have an effect, but hopefully right now it's not having it that much. We still don't know whether this is underreported cases or not. And the other point that you touch is the vaccine. So we don't have a vaccine. There's only one vaccine that I think has been European agency has approved it, but it's a very bad vaccine, only 30% of accuracy, and protection wanes very, very, very quickly. And this probably Freddie can cover more on that. But this is because malaria parasites are so complex and so diverse that it's very, very hard to find a vaccine strategy that really prevents infection.
It's if I understood correctly again, it's also something to think about if what you're sharing with people is actually there are effective therapies available, but still hundreds of thousands of people continue to die every year. Mm-hmm. It's a lesson or a message about having an inclusive world health view of how we treat these things. And we're if we come back to the current pandemic, I mean, we're thinking about how to develop vaccines or antiviral compounds, but how do we share them with the world? So it's not just weaponized so that some countries are protected and others aren't. Freddie, maybe you have a comment you can add to that.
Yeah, thanks, Angus. There's obviously many things you can do. So maybe very quickly for those who don't know me, so I did my PhD with Michael Way at EMBL in 96 to 2000, working on vaccinia virus. So pox virus that was used as a vaccine to eradicate smallpox, as Raffaele mentioned. And then I thought, well, that pathogen is already eradicated, so let's work on something that's maybe more interesting. And I thought about bacteria, but then I thought, let's work on the most complicated thing, and I went to work with parasites. And now I'm back in Heidelberg, and since 15 years working on on malaria parasites, but how they're transmitted from the mosquito to the human, or actually mouse host. And having to teach, of course, I teach parasitology a lot. Malaria is just one of those many parasites. There's about equal numbers of infected people from schistosomiasis, which is a debilitating disease as well. It doesn't kill as many as malaria, but it's in health terms, it's probably just as bad. There is probably in the range of 1 billion people infected by hookworms, which is also debilitating. In some countries, there's 20% of school kids have worms that are soil transmitted. Things we just don't really think about in our daily lives, and that really contributes to them not being able to go to school. Just things that happen now to our kids as they can't go to school and don't get an education. That's the unfortunate status of daily life in the third world.
Freddie, can I ask, as an expert in understanding much better than me politics and science and society, do you feel the allocation of resources are being driven more these days by social media than by actual fact-driven, thought-developed processes by our politicians? In other words, the impression I have is our politicians increasingly respond to fear of social media campaigns, and this can push them to make short-term decisions about how to allocate resources, what to do. Should you lock down? Should you not lock down? Should we try to get a vaccine? Do you think that's distorting the way that we use science and make decisions collectively?
I would say the short answer is yes. But that's not a new thing, because look at the HIV pandemics, right? This was driven by people who were sick of certain groups and who made massive pressure to the governments, and then there was massive amounts of money being put into HIV, and it still is. There's still more money in HIV research than in malaria, even during the HIV pandemic, more people have died cumulatively from malaria than from HIV. But this will always be the case. I think that that's politics, right? That people will shout, and the one who shouts the loudest will get money. I'm not sure I have other than saying yes, that's true. I'm not sure whether as a scientist, I think this is a this is a totally bad thing, because that's essentially the fundament of our democracy. And one would hope that it would be less of a fear-driven argument, and it would be more of an information-driven argument to allocate resources. But I think what this pandemic, to me, drives really back, or what I hope it drives to other people, is that we're limited in our resources, and we should maybe be very careful of how we spend our resources. Right, the resources are not limited; they're not unlimited. Right. And when we just decide to spend something in one thing, we just don't have the money to spend it on something else. And of course, my favorite example is the military, right? I mean, how can we even talk about whether we should put more money into malaria versus COVID versus HIV versus something else when, we spend, I don't know how many trillions of just total wasted money into people running around with guns and building rockets and all this crap.
I mean, one way
that gets into a discussion that I'm not sure we should have today.
We can absolutely have the discussion. I'm not disagreeing with you, but in terms of getting the maximum value from the money that is spent by the taxpayer on research, including in the EMBL, which is primarily money from European taxpayers. I mean, my own feeling is that we need to improve even further the culture of community-based science. By that, I mean focusing more on sharing results and data than simply using the resources to compete in as if it was the Olympic Games of science, where the most important thing is that people use money to advance individual careers and win prizes and medals, as opposed to use the money that society is donating to learn more collectively. So maybe that's an unrealistic ambition. It's a slightly different direction to the one you're indicating, but maybe it just reflects I'm now old. I'm less worried about my future career. But if anyone else would like to comment as to how you can achieve that by still allocating resources based on somehow rewarding performance without making it overly competitive, so that we lose value by having people wasting money doing the same thing or racing to do the same thing. I don't know if you have any thoughts on that, Freddie, or if any of the other.
You could just get back to the COVID question that you asked, right? So I did think whether I could contribute to COVID. Lots of people in our institute did. Ralph Bartenschlager, who was instrumental to develop actually hepatitis C treatments and narrowly missed the Nobel Prize, unfortunately for us in Heidelberg. He's at the institute. They did lots of corona research together with EMBL people, Martin Beck, John Briggs, EMBL alumni, and they published wonderful papers. And I thought whether I could contribute, but I quickly realized I can't, simply because we also didn't have the resources in terms of rooms available to do the research. And at the same time, I think, I would have not spent a single euro on public money on corona vaccines, because it was very clear there was enough money from people who were having money and who would be taking the risk of, investing this and then getting some risk investment back if their vaccine moves. That's probably a controversial thought.
So one other comment I know that we've discussed before, Freddie, is that in the UK where I'm living, I'm continually hearing locally and nationally from politicians who say all the responses to the current pandemic are being driven by what the scientists tell them. And yet I'm extremely aware that the advice they're getting at the moment isn't uniformly the advice of all scientists. And in fact, there are some quite divergent opinions about how to tackle the same problems. And I think it's a problem that the governments tend to cherry. I worry that the governments cherry pick which scientists they want to listen to, and the media also doesn't give a sort of voice often to people who have different points of view. I mean, is the same situation happening for in Germany or in any of the other countries for people taking part? Do you feel that
here in Italy, and it is a pity because actually, let's say I think scientists lost a chance to actually being very popular here because of this reason. In other therapeutic areas that I work with, like hepatology, every year there is a European consensus consensus conference, which is very useful because, guidelines are developed for governments and everybody. So I think we need something like that in the COVID-19 arena as well. We need consensus statements from the scientific community. We don't need newspaper articles or even worse, tweeting or putting things on Facebook from scientists. That is going to be just raise controversies.
Yeah, absolutely. I figure that's a point. So once again, an hour has flown past. And I want to invite everyone who has time to stay on for a second cup of coffee with EMBL and to use the opportunity to let anyone interested in contributing. I've seen some really interesting points being made in the chat. Particularly, I've picked up on some discussions that I think are extremely relevant as to why there are such variations in the number of apparent infections and the number of morbidities resulting from those infections across closely related countries and even different areas within the same countries. And I'm sure a lot of people here could contribute to that. But after the first hour, I just want to make a point of thanking all the participants, and I hope they'll stay on, Raffaele, Jan, Maria, and Freddie for the great contributions again today. And just to remind everyone, before we finish after the second cup, that there will be another session on Friday, the 13th of November. If you dare to come along on that date, we really look forward to seeing people again and see how this situation has evolved. I really hope we are celebrating better data and more freedom of movement and less restrictions. Personally, I'm not optimistic in 1 month's time that's going to be the case. Noosh, is there anything else you'd like to mention at this point to share with the community?
Not really. You've covered everything. I also look forward to Friday the 13th with the community, loving the fact that everybody's joining in. And if people have other aspects, relevant topics they'd like us covered, we have obviously some speakers in the back end that we're in discussions with for the planning. But if you have a specific need or you're seeing a trend, just write to us with your ideas. Alumni EMBL.
That's fantastic. So, Raffaele, do you feel that do you feel that the politicians have an accurate view of what's realistic to deliver in terms of vaccines or antivirals? Because whatever we may think of them, our political masters are mostly not stupid people, and they're obviously, but they're mostly not scientists, and they're responding to information that's being passed on to them. Do you feel that they've got an accurate view of what's realistic to expect?
I think that not even scientists have a homogeneous, realistic view of what to expect. So I think that scientific community should really come to a consensus. But in terms of vaccines, for example, I was just reading on the news that Pfizer has announced that they're going to be a vaccine licensed by November, which is probably, not impossible if they have good phase 3 data, maybe a bit rushed from my point of view. But again, I think that having a vaccine license is different, very different from being able to vaccinate most of the people who are in need of a vaccine. So that message should be, even some investment at risk was made in terms of manufacturing plants. I don't think that it's going to be possible to manufacture all the vaccine that is needed. And there are questions about safety. Now, the other thing that I want to raise is that even, probably a vaccine with 50-60% of protection would be approved. Now, face masks gave just about the same level of protection. So I think we should really use them heavily and keep this in account. So even if we have a vaccine of that sort, is not going to be enough. We probably will have to combine it with wearing face masks for a little while at least.
I'd like to bring in Toby Gibson, who many of you will know from EMBL in Heidelberg, who I know is interested in looking at antiviral candidates. Toby, do you want to contribute now?
Yeah, I absolutely do. I was thinking a little bit about what to say since I don't join this. First thing is that I'm older than Angus, and everybody who's older than Angus is high risk. Everybody who's younger than Angus is low risk. So, of course, being a bioinformatician and working from home office, I can spend a lot of time looking at the COVID literature. And the first thing I want to tell people is that although everybody talks about the ACE2 receptor as being the receptor for the virus, it might be in some tissues, but it's not in the lungs. If you can see my screen, I can show you the human protein atlas. Tell me if you can't see it. Human protein atlas is made by an alumnus of EMBL, Matthias Ullén. And the lungs...
You may have to share your screen again. At least I can.
I'll see it at the moment, and then I'll be there.
Okay, now...
And there isn't any protein in the lungs and a tiny bit of RNA. If you stay individual cells, you get a tiny bit. This other website from researchers in Nottingham has also got a lot of ACE2 lung and patient data. So these are very important. There's also publications, several different publications, all agree, but there isn't. Now, there's a 2004 paper with an enormous amount of ACE2 in lungs. It's an active paper. Receptors, and the two main ones are neuropilin and interins. And I can talk more about that, but I would like actually to talk about drugs. So, as mentioned today, Gilead's drug is not working, remdesivir, for the World Health Organization, apparently. There's a simpler version of that drug that doesn't have a phosphate. That means that it's absorbed more quickly into cells. Gilead don't want to pursue it because their patent is older, so it would expire sooner. The cats get a coronavirus disease. Rather like the COVID, most of them get better, and a few go into a chronic illness. And then their cat owners get sad, and in America, what they do is get that from people.
I think we're losing you, Toby, with this internet connection.
The websites, but I'll show them that page, so I can probably stop showing my screen. So the simpler version is probably less toxic, and it cures cats of chronic disease in America. When they it doesn't cure all of the cats, and of course you're relying on getting an illegal shipment of a drug, and it can be anything. I can provide links later on to put into the thing, but that's the NIH are now going to check that drug, and it might work better than the phosphorylated version, which can't get into the cells until the phosphate's been taken off. So that's one story. There's another interesting drug which is called Ephrussi leukemia, almost all old, so they're high risk because they're cancer patients. That's a drug that's licensed, and so CLL patients get Ephrussi. And of course, when some of them got COVID, the doctors are very worried that they were going to die because of immunosuppression. Because what the BTK tyrosine kinase enzyme does is it's in all the white blood cell lineages, and therefore it will suppress B cells, immunity, and so on. However, it appears that the CLL patients were protected from getting bad lung disease, and there are several papers on this from independent people all saying the same thing. There's another drug called Ephrussi that's not yet licensed, but is under trials, and there's also a paper saying that that is protective. So this is not about curing the virus. This is about reducing inflammation by macrophages into in the severe lung disease. That's a very interesting drug. And then my favorite is a chemical POSH. And it's an antipsychotic drug called chlorpromazine, and there's a family of them, tricyclic antipsychotics. It turns out that virologists use this in order to see, in tissue culture, to see if viruses are using endocytosis, which coronaviruses, by and large, are using. So you put it in a tissue culture and it inhibits endocytosis, and then you can say that my virus is going through that system, or it's not going through that system and it's not affected. And there's going to be a drug trial in Paris of this, because they observed in a psychiatric hospital that anecdotally 4% of patients got severe COVID and 15% of the health staff got it. So I think
one of the things that occurs to me as being a serious problem, Toby, is great that you can highlight these interesting candidates and just to unearth this. I mean, I guess one of the problems, maybe Raffaele again could comment on this, but the trajectory and timelines that are normally involved in drug discovery pipelines are too long for allowing a rapid response to a situation like the one we currently are in. And I think that needs some sort of paradigm shift. Also, there's the danger that you rush to take premature signs of a positive result and rush that through, and then it turns out that actually the data on closer inspection don't hold up. I mean, I don't know if you saw this, Raffaele. I saw in the news today that one of the first drugs that was supposed to be helpful in improving survival rates, actually a deeper study showed actually had no real benefit. Was that just in the UK, or
is that remdesivir, the Gilead drug?
Yes.
Which the one that Toby mentioned also. Well, I think one of the issues with that sort of drug with all direct antivirals is that if you test it on on people who are advanced disease, they're not going to work. You need really to test them right after when you get infected, and that is difficult to do because But essentially, if a window of opportunity for a direct antiviral drug is only a week or so, so we have if we want to really control the virus with direct antivirals, we need to be able to test a lot of people and very rapidly, and which we're not right now. So that is an issue of the way clinical trials are designed. But even if we have a drug that works, yes, it will take time before we actually show that it works. I mean,
yeah.
The same is true for vaccine. For example, we talk about vaccinating first people, like older people who are at more risk, but most of those trials are not being run on older people, but they're being on young, healthy people, and so we are maybe like the vaccines, drugs and vaccines are approved, are licensed with indications. So maybe first wave vaccine will not get indication for people who have comorbidities or older age. So it's going to take time. Yes.
Yeah. And Carmen in the chat has also pointed out correctly that there must be opportunities for repurposing existing drugs that have already passed clinical safety trials. Freddie, could I bring you back in again, perhaps to comment on one of the other things that must be a big part of this whole response in society is testing, efficient testing. And certainly where I live nationally, that has not been an unqualified success. I hope that's a politically neutral enough statement that it's not going to freak out the EMBL, but I think it's safe, and most people in the UK would agree it's not been an unqualified success. I'm actually involved at the moment in trying to help colleagues set up an on-campus to take on this one, to doing it ourselves, and just to set up a system for testing this on campus ourselves for our colleagues, independent of the government-based system. But I mean, it seems to me in Germany's done a pretty good job of this so far. Are there lessons you feel that other countries could learn from how you've gone about it, or anything that, why are you doing it so well?
Well, first, I would say it's not because we're particularly well organized, as some people might expect, but I think it's got to do with the federalism that happens here in Germany. So that a lot of local clinics actually just went in to develop their own tests and applied whatever test was available, and they were allowed to do so. Well, the way I understood is that in the UK, this was very much National Health Service driven. So they wanted to have one thing and then make that one thing go through the entire country. So, like Michi Knob, an EMBL alumnus who was a group leader at EMBL, he developed a LAMP-based assay, a rapid assay, and he's currently, they published it in Science Translational Medicine a few weeks ago, and they now are trying to basically get this to the level where they can do this to do high throughput testing. And so I think for currently, if you sign up on our campus, you can get just tested. It's not an approved test, so it doesn't officially count if you're positive or negative, but you know for yourself whether you should be careful or whether you can maybe be a little bit more risky in your daily life. But I think that's the key thing, and I think that's maybe a lesson one can learn, that we tend to always create bigger and bigger structures. It might be a lesson for EMBL. Maybe EMBL shouldn't become bigger and bigger and bigger, because at one point it might not work anymore that well, right? I think there is a certain beauty in having small units and allowing these small units a certain level of freedom. Right?
It's interesting. Yeah, I think
that's I think what really made Germany somewhat successful.
Yeah. Maybe I could say something about the NHS. It's obviously the NHS has been badly run generally. The very interesting thing is that they can run very good trials, and I think that's, they can run proper clinical trials because they can essentially organize that and put it across multiple hospitals. And you can see that this is a really useful piece of work they've been doing. They did the dexamethasone trial, for example, and they did their own chloroquine trial and things. Almost everything else about the NHS is a disaster. They don't have enough beds and things because they're deliberately underfunded and outsourced and things. But it wasn't really.
I mean, these are obviously complicated questions, and I think sure. I'm not disagreeing with anything you said, although I remind everyone it doesn't represent the official view of EMBL or any specific member government. In case this is communicated. It isn't only about spending money. I mean, that's not to disagree with you, Toby, but I think understanding how to respond to I think Freddie actually raised an incredibly important point that bigger isn't always better, and perhaps finding more effective ways of doing things, and maybe the same organization that's very good for one set of purposes, perhaps treating people with chronic illnesses or not life-threatening illnesses, maybe isn't the same structure that's best placed to respond very quickly to severe life-threatening situations. I mean, I think it was very interesting that Raffaele had pointed out that all this work in development on antivirals, maybe you could get as much benefit from more people wearing face masks or some other action that would be faster and cheaper to implement. If I remember correctly, and perhaps other people could correct this, actually moving historically to having clean water for the population had a bigger impact on health throughout Europe than any other therapeutic development. Just giving people clean drinking water was probably the biggest single contribution to improved health. I don't know, Raffaele, if you want to comment on any of these points.
Sure. Obviously, yeah. And I think, again, face masks work pretty well. I mean, and again, some of the vaccines aim at having similar results. So we have to bear that in mind. It's just that people don't like to wear face masks. They want a vaccine because they want to stop wearing face masks, which becomes a political problem. And but yeah, so we have to keep in mind that I personally think that we can really, between using precautions, being careful with distancing, hygiene, and face masks, we can really prevent more than 90% of infections. I mean, basically, and I don't think strict lockdowns are necessary. They may have been in the past, but I don't think we can use strict lockdowns as a preventive measure and not do anything else, because if we lock down for the next 1 month or so, then in January we have the same problem that we have now. So I really think we have to learn how to live with COVID-19, and it's not going to last forever. It's not last another year or so, and hopefully protect the people who are more at risk with vaccines whenever they're rolled out. But we are not going to be able to leave all the other measures, even after vaccines are licensed. And for antivirals, it will take longer for sure.
Raffaele, do you think that, or was there evidence that initial infectious load is very important? In other words, that if you get a very large dose at the beginning, that you might get a very severe illness.
And that is very, I don't think there are data already out to actually validate that hypothesis, but I think that is very reasonable. I mean, for at least in animal models, for a variety of viruses, the more virus you give, especially for acute viruses, and the more damage you do all the way to death. There are data in flu vaccine trials where people were subjected actually to a volunteer infection that show a very nice correlation between dose and severity of a disease for a respiratory virus. So that may very well be the case for SARS-CoV-2, and maybe one of the reasons why the second wave causes less mortality and morbidity compared to the first wave is that, at least in Italy, but I think in many other countries, transmission of the first wave of SARS-CoV-2 was actually took place in hospitals, in very crowded places, in nursing homes, and probably the dose that people were exposed initially was very, very high. Also, where people were not wearing masks, WHO said masks did not help, and anyhow, they were not available for most workers in hospitals, and so I think that may be a real thing. And there is even somebody has hypothesized that actually we are getting sort of vaccinated by wearing face masks because you get low levels of virus and you develop a relatively good immune response rather than a nasty one, and by getting exposed to low amounts of virus, and that could be beneficial at the end. And I think it's a possibility. It's a very plausible thing. I don't think it's been scientifically proven yet. But it's
Raffaele, a question came in for you in the chat. I'd like to give you a chance to respond to from Robert in Exeter, who wanted to ask. It sounded like you're very confident that coronavirus will not be around in another year or so. I assumed you were referring to like historical precedents from other pandemics or recent experience with things like SARS. But was there some other? Do you have other reason for confidence in this view?
No, not really. I mean, it's based on history. I mean, even the 1980 pandemic flu had an initial wave, which was smaller than the second wave, and then we were smaller waves at the end, but less mortality and morbidity because by further waves as herd immunity was developed. Now, I mean, obviously, I don't know for sure. I mean, it's just my guess would be that we may have. I mean, we're going to probably have a problem this winter. And I think this virus is trying to become seasonal. I mean, it's not there yet, but eventually it will probably become a seasonal virus like other coronaviruses. It will take time. I don't know. My guess is that in terms of people being affected in a bad way, morbidity, mortality, my guess is that the worst has maybe in time we'll see another wave, and I hope that subsequent waves will be gradually smaller and smaller. But it's just maybe a bit of wishful thinking, or I hope so. But that's just my gut.
I think I'm going to take that as an optimistic note, Raffaele. I said at the start that I wanted you to leave us all feeling positive that we were all in a better position, that things were going to be okay. So I guess that's maybe as close as we're going to get. I've really enjoyed seeing so many friends, meeting some new ones. And again, I'd like to thank all the speakers from today, Raffaele, Jan, Maria, and Freddie. Thank you very much. Thanks to everyone. It's been a great discussion. I've learned a lot, and I look forward to seeing everyone again soon. And I wish everyone all the best. Bye-bye.